So our next class topic is going to be on proteins and protein structure. The topic of this video is amino acids, and amino acids are the building blocks of proteins. And to give it a little bit more of a fancy terminology, proteins are polymers of amino acids. And a polymer is a chain of repeating subunits. And we call those subunits monomers. And quite a few of the biological macromolecules that we're going to talk about in this class are actually polymers. So they're long chains of a repeating monomer or subunit that's linked together by a particular kind of bond. For proteins, the monomer is amino acids, and amino acids are going to be the focus of the video today. And it's really, I think it's really important that you be very familiar with the structure of amino acids, and even to the point of being able to draw it. Because that way, when you see an amino acid on an exam, you'll recognize instantly what it is, and you can kind of key in on the pieces of the structure that might relate to the question. Drawing is a really, really good way to learn. So the structure of an amino acid, well, it starts with a central carbon like this, and then it has a functional group on either side. On one side is a carboxyl group, and we've learned about those in class recently. And on the other side is an amino group, which you may remember looks like that. But our amino acid isn't done yet, because notice that the carbon only has 2 bonds, and it needs to have 4 for it to be fully bonded. So it has a hydrogen up top, that's kind of a placeholder, and then it has what we call an R group. So I'll just write that. R group. You also see this group sometimes called the side chain. These are just different terms for the same thing. And I'll get back to the side chain in a moment, but let me label these other groups here just as review. So there's our carboxyl, and then here's our amino. And remember that a carboxyl group is an acidic functional group, so it has a tendency to lose a proton or lose a hydrogen into solution. It loses this one right here. The amino group is a basic functional group, so it has a tendency to pick up a hydrogen. And so sometimes you'll see that amino acid drawn a little bit differently. Sometimes you'll see it drawn like this. And I'm just going to kind of draw this quickly. And with a plus there. And so this is also an amino acid, but it's in its ionized form. Ionized form. This is what a free amino acid would actually look like if it was in solution. Those, the acidic group would lose its proton, the basic group would pick one up. But you can see it's the same amino acid, and it even has the exact same chemical formula, because it's just lost the H over here and it's jumped on over there. So don't be thrown if you see an amino acid presented in its ionized form. And honestly, I can't remember how your book does it. I don't remember which it shows. And the structure of these R groups varies among amino acids. I'm going to tell you a little bit about that. I'll just point out that I took a cut to erase the board over there because we're going to need that space in a moment. So the R groups can be, they can be nonpolar. They can be polar, acidic, or basic. Those are the categories that they fall into. Nonpolar amino acids are, nonpolar R groups, and those all fall into the category of nonpolar amino acids. They tend to have R groups that have a lot of carbon, hydrogen bonding in them, carbon-carbon and carbon-hydrogen bonds. And remember that those are non polar covalent bonds. And those can make, they can make chains, they can make branches, they can make rings. So there's a number of different structures that fall into that category of nonpolar amino acids. And there's actually 10 of the amino acids that we use that are used to make proteins are nonpolar. Polar R groups tend to have polar bonds in them. So they tend to have things like carbon-oxygen bonds or oxygen-hydrogen bonds. So either carbonyl groups or hydroxyl groups in there. And they have partial charges. So either a partial negative or a partial positive. The acidic amino acids have a full negative charge. They tend to have a carboxyl group. So remember, the carboxyl is COOH. I'm writing it this way. This is a picture of what it looks like up here. And they tend to have a full negative charge to them. So remember that something that's acidic loses protons into solution. A carboxyl group loses its H into solution, and that gives it a full negative charge. And there are, sorry, I kind of got behind here. So there's 5 different types of polar amino acids. There's 2 acidic amino acids. And then finally, the basic amino acids have amino groups on them, or they have some kind of a version of an amino group on them. And so they have a full positive charge. Remember that amino groups are ones that tend to pick up a proton from solution, and there's 3 of them. So if you add that all together, you get 20 different amino acids that all fall into one of these 4 categories defined based on the structure of their R group. And I don't really have room to draw you examples of those, but there you can find a whole page in your book that shows you what all those structures look like. Okay, so the next thing I want to move to is how these amino acids are linked together to make the polymer that is a protein. And they are linked together through peptide bonds. And I'll draw what that looks like down here. So I'm going to draw a couple of amino acids. Let's see, here's our N, HH, C, C, O. I'm going to draw it in, I think I'll draw it in the ionized form. And there's our H, and there's our R group. And that's going to be plus, and then we've got another one here. Okay, and so they get added together. So 1 amino acid plus 1 amino acid equals, so here's our N, H, H, H. There's our our alpha carbon, our essential carbon. Okay, now here's where things get a little bit, this is where the peptide bond is formed. And what happens in the peptide bond is this oxygen and these 2 hydrogens actually come together to form water, H2O. And what's left behind is now a new bond between the carboxyl carbon and the amino nitrogen. And it looks like this. There's that double bonded O on the carboxyl hydrogen. There's our nitrogen, and it's got 1 hydrogen left, and I'm going to draw it down here because that's actually how it would normally be oriented. And then there's our carbon, there's our H, there's our R, and then there's the other carboxyl group. Okay, so the peptide bond is the new bond that was formed is this guy right here. So there's our peptide bond. And the kind of reaction, this type of reaction has a name. It's called a condensation reaction. Let's see. So a peptide bond is formed through a condensation reaction. And it's called condensation, and I'm gonna have reaction as RXN. A condensation reaction is called condensation because water is formed through the reaction. So every condensation reaction shares that characteristic that water is formed. A peptide bond is it's a covalent bond, and it's between the amino N of one amino acid and the carboxyl C of another amino acid. And I'm not going to write amino and carboxyl because I'm kind of out of room, but that has to be true. If you're joining two amino acids together, it has to be the amino N with the carboxyl C because it can't bind with the C that's in the middle of the amino acid. So it's a covalent bond between the N of one amino acid and the C of another amino acid. That's how you link them together. And peptide bonds are really strong, stiff bonds. It takes a lot of energy to break a peptide bond. And I know this, I have personal experience with this because I work with amino acids in my lab. And when we start with a protein and we want to break it down to amino acids, we have to put it in extremely concentrated hydrochloric acid. We cook it in an oven and we leave it in there for 20 hours. So we have to apply heat, time, and very strong acid to get those bonds to break. Of course, in your body, that's not necessary. You have a little enzyme that just comes along and clips them off. We're going to talk about enzymes in the next class.